Thank you, and also thank you Danone and Paul Sheets for having me here in Paris to talk about food as medicine. And we live on a hungry planet. We can all agree on that. And I find these pictures here extremely interesting. This shows what a family eats during 1 week in different parts of the world. Look at the family from Louisiana in the upper left corner. Compare what they eat to the family in the lower right corner, a peasant family from Ecuador. It's like they are living on different planets. And of course, this will have a major impact on health outcomes. And of course, food is just a bunch of molecules. We can all agree on that. And when you make your dinner at home, this is advanced chemistry in your kitchen you are dealing with. And I think this is what makes nutritional research so very interesting. This is advanced chemistry. The foodome is a discipline that studies the food and nutrition domains through the application and integration of advanced omics technologies to improve well-being, health, and knowledge. And this is so complex because the foodome includes more than 25,000 different substances that make up our diet. And of course, the aim of this foodome approach is to link the diet to our eating patterns to the individual genome and the individual diseases that accumulate as we get older. This is a very important piece of paper. It's a review summary published in Science 2021, discussing the fact from fiction when we have all these anti-aging diets that are now so popular. We have the caloric restriction diet, we have the time-restricted feeding, the protein restriction, the ketogenic diets. And as discussed in this paper, validated biomarkers of biological aging are required to match intervention to each person's distinct genetic and environmental context and thereby optimize the individual healthy lifespan. And I think Paul addressed very well that we now seems to have an opportunity to use epigenetic clocks as biomarkers to study the effects of these anti-aging diets. Me and Paul and other dietitians, collaborators in the renal field, we have since many years been interested in if we could use food as medicine to target this very toxic uremic phenotype that we see in the patients that I care for, those with chronic kidney disease. And this is based on a review we published in Nature Reviews about different possibilities here. One possibility that I would like shortly to address is to target transcription factors that are involved in inflammation and oxidative stress. And when we went through the literature, there is a plethora of opportunities here. We have, for example, sulforaphane, which I will discuss a little more in detail. We have curcumin, anthocyanins, quercetin, flavonoids, rhizome, and cinnamaldehyde, and many others. And the basis for this is, of course, what Paul already discussed, this diseasome of aging, the burden of lifestyle diseases that accumulate when we get older. They are, some of them, most of them are depicted here. It includes not only chronic kidney disease, but of course only fatty liver disease, heart failure, Alzheimer, obesity, atherosclerosis, osteoporosis, periodontitis. And these are seemingly very different diseases, but they have two things in common that I find extremely interesting. The first one being this chronic smoldering inflammatory response that may accelerate biological aging. You know that this is called inflammaging. We also have this association between inflammation and the loss of gut biodiversity. And I think these are two very, very important factors that we could target if we want to lower the biological aging procedures associated with the diseasome of aging. I actually spend 1 week of my life to go through the literature to find out what has been published regarding NRF2. that Paul mentioned in these burden of lifestyle diseases. And it was quite obvious in the Friday afternoon after this extensive review, all this paper, that irrespective if you go into patients with obesity, atherosclerosis, Alzheimer, heart failure, fatty liver disease, diabetes, depression, cancer, periodontal disease, in general, the expression of this transcription factor was reported to be low in different tissues. And this kind of evoked my interest in this transcription factor. It's actually a two-component system with NRF2 and its inactivator KEAP1. And if this is activated, there will be an upregulation of hundreds of cytoprotective genes that is known to reduce oxidative stress and inflammation. And what I found really interesting, that this transcription factor is something you can affect by your lifestyle. We know that smoking, we know that psychosocial stress, the Western dietary pattern, and in my specific field, uremic toxins downregulate NRF2 expression. Exercise is something that upregulates it. And then we have a number of pharmacological synthetic NRF2 stimulators. Cloto is one of the maybe most important, but then we have drugs that we commonly use that has been shown to stimulate NRF2. Metformin, a drug with many important effects, statins and lithium are other drugs that may stimulate NRF2. But for the audience here this afternoon, I think the effect of bioactive compounds and fermented food on NRF2 may be the most interesting. And indeed, again, going through the literature, many studies show that on nutrients like kimchi, curcumin, rosemary, tomato, coffee stimulate NRF2. Actually, the most potent stimulator of NRF2, it's not broccoli, but broccoli sprouts, sulforaphane. And there are now studies that have shown that actually sulforaphane have quite potent effects. And this is a study published in Science Translational Medicine by a Swedish group. They could first show in the basic science part that with increasing doses of sulforaphane, there is an activation of this nuclear transcription factor NRF2. But more interesting, I think, are the clinical findings, because they studied the effects of broccoli sprout extract in patients that are obese with dysregulated type 2 diabetes. And as you can see here, there were quite marked effects on the fasting glucose and glycated hemoglobin. More importantly, they could actually show that the hepatic glucose production decreased in the same magnitude as you will find with metformin. There are many other studies on sulforaphane. I have no time to show them here. This is one study showing that sulforaphane actually have effects on non-alcoholic liver disease. And I just like to mention the recent study in Nature Microbiology from the same Swedish group that showing that the effects of the broccoli sprout extract is very much dependent on your gut microbiome. So you may have different effects dependent on your gut microbiome, which seems to play a very important role if you will have clinical effects of sulforaphane or not. I work in nephrology, and we work on uremic toxins that accumulate when renal function declines. And there is now emerging links between the gut microbiome and the cardiovascular disease mediated through these uremic toxins. We have short-chain fatty acids that have effect on blood pressure, myocardial repair, cresyl sulfate, indoxyl sulfate, the lipopolysaccharides. We have phenylacetylglutamine, PAG, which have the documented effects on atherosclerosis and thrombosis. But last but not least, I think the most interesting ones is the link between trimethylamine N-oxide and atherosclerosis, thrombosis, and cardiorenal fibrosis. So TMAO is produced from food rich in choline and carnitine. It's atherogenic, prothrombotic, and inflammatory. We know that red meat is particularly rich in carnitine. In addition, red meat shifts the gut microbiome to produce more of the precursor of TMA. And on top of this, red meat actually reduces the ability of the kidneys to excrete TMAO. So the patients I care for, they have extremely high TMAO levels. And we just learned from this paper published in a respected renal journal, based on more than 10,000 participants from 2 community-based cohorts with a follow-up of almost 10 years, that high TMAO levels were associated with a higher risk of incident chronic kidney disease and a greater rate of renal function decline. And what struck me the most is that the association with a decline in eGFR was similar or even larger than that seen for the established risk factors that we care for so much, including diabetes, increased blood pressure, older age, and also black race. This implies that we actually now have an emerging risk factor that we need to take care of a lot. And it has been discussed, like in this paper in JAMA, that TMAO may be the new red meat risk factor. When I give talks about TMAO, one of the questions I usually get, how do you lower your TMAO? Or dietary interventions may be the most appropriate way to do this. I just like to mention here this 3,3-dimethylbutanol. It's a natural compound you find in olive oil, red wine, and some vinegars that has consistently been shown to inhibit the formation of TMA. We also have resveratrol and other polyphenols that also suppress TMA-producing bacteria and lower TMAO in preclinical studies. So there are ways to reduce your TMAO levels. But the most important factor is, of course, to be considered what you eat. And red meat on the top here has carnitine. We know that carnitine in the gut is formulated to TMA, which in the liver will be metabolized to TMAO, which have all these toxic effects. There is no doubt, if you compare to white meat or a non-meat meal, eating red meat will increase your TMAO levels quite markedly. Then we have egg. Egg is rich in phosphatidylcholine, and choline, which may also via TMAA be metabolized to TMAO. But this is a study that actually showed that healthy participants eating 18 eggs per day, there was no rise in TMAO. Compare this to the choline tablets and the phosphatidyl or the egg whites with choline tablets, and this is good news for people like eating egg like me. No rise in TMAO. And then finally we have fish, and fish is something that we usually consider to be healthy for. So there is an interesting fish TMAO paradox, because fish is rich in both TMA and TMAO. So how can this paradox be resolved? Well, to explain this, we really need to know why TMAO exists in nature. It's something that counteracts the effect of these stabilizers, such as low temperature, high urea levels, or high hydrostatic pressure. In fish, the weight of the water will push water molecules into protein and distort them, and fish needs protection for this. And we have really learned a lot from this extremely ugly fish, Notoliparis kermadesensis, which is the second deepest fish ever seen alive. And this paper in PNAS actually show a very strong correlation between the TMAO levels in the fish and the depth at which the fish were catched. So you usually have a higher muscular TMAO content in fish habitating the deep sea. And this may have actually an impact. So Stanley Hazen's group, they actually examined the concentration of TMAO and TMAA. That is the red bars here, and also the eicosapentaenoic acid, the fatty acids, the blue bars in different fishes. And I find these results extremely interesting. You can see here on the right side that you have some sea creatures such as cod, lobster, halibut, which have high levels of TMAO and TMAA. And on the left part, you will find shallow living fish, which have low or even non-existent levels of TMAO. You have, for example, the trout. You find in shallow waters with very high levels of eicosapentaenoic acid, but absolutely no TMAO. So deep sea fish have high content of TMAO, and shallow living fish have low or non-existing TMAO levels. And this will have an impact, because here you're giving healthy controls a diet of shrimps or tuna, absolutely no increase in TMAO. But look here when you give them wild salmon or fish sticks. Fish sticks, I think, is usually based on cod. And this is what we like to serve our children in school, fish sticks for lunch. And you can see these extremely high levels of TMAO after eating this meal. So based on these findings, we studied 824 older patients with advanced kidney disease, and we followed them for 4 years. We studied TMAO levels, reflecting the intake of red meat and fish, but we also evaluated levels of carboxymethylpropylfuran propionate, which is something that reflects the intake of the fish and fish oils. And then we did something very simple. We just divided these patients into 4 groups based on the median levels, and then we had one group with high TMAO and low CMPF, and this would imply the high intake of red meat but low intake of fish. And when we followed these patients for 4 years, we found that this group, they have a higher risk for all-cause mortality, but specifically extremely high risk of incidence of kidney replacement therapy. They needed to start dialysis, indicating that this diet with red meat and low in fish is something that accelerates kidney decline, confirming what was shown by Stanley Hazen in the other study here. So here I think we have one of the most important emerging risk factors for kidney disease. So we now know that the link between what we eat and organ function is with the microbiota. And here I think this is an extremely interesting review by Erika and Justin and Sonnenberg discussing these links. And the problem we now face in this era of the Anthropocene, which we now live in, with the industrialized microbiota that actually has never before been experienced by our race. So having a healthy gut microbial biodiversity is like having a rainforest in your guts, is something that we really want. And we know now that fibers, fermented food, coffee, beer, exercise is something that stimulates our gut microbes. But more concerning is that more and more of us will have a poor gut microbial biodiversity. We have palm tree plantations in our gut. And what's now happening in our planet with increasing in temperature, with air pollution, artificial sweeteners, salt, unnatural environment, not to say the least, the microplastics will actually affect our gut microbiome and lower the diversity. And of course, the toxic food that we like to eat so much is maybe the most important factors that affect our guts. And looking during the time of humans on our planet, there has been a major dramatic change in our microbiome from hunting and gathering period to the ancient agriculture period to our now the modern Western diet with a loss of biodiversity, loss of function due to the toxic food we like to eat, and not least all the pills and drugs that we eat. Proton pump inhibitors have dramatic effects on our gut microbiome. So maybe there is something missing in our diet now, and I think this was an eye-opener for me, and I discussed this paper a lot with Paul here about the activation of the NRF2 cell defense pathway by ancient foods. Something that is something we have lost in our modern Western diet. And something that we really don't eat much now, at least in the western part of the world, is fermented food. I know in Asia, Korea and Japan, you have kimchi, you have natto. In Germany, you have the sauerkraut. But with the fermentation, you will actually invite the microbes you want, and you don't let in the ones you don't want. And the importance of fermented food, I think, was best shown in this paper again from Justin and Erika Sonnenberg's group. It was published in Cell 2021. They could actually show that a high-fiber diet changed the microbiome function and elicits personalized immune response, but more importantly, that the fermented food diet increases microbiome diversity and decreases the markers of inflammation. I think we need more fermented food, at least in the western part of the world. Fermentation increases digestibility, it improves organoleptic characteristics, it provides prebiotics, probiotics. Abundance of research show that the fermented food has positive effects on the gut microbiome. It stimulates NRF2, it has inhibitory effects on NF-kappa B, and in this will lead to immodulatory effects that decrease inflammation, oxidative stress, and this could have benefit from many of the burden of lifestyle diseases that we have problems with today. And this could be actually a way to reintroduce evolutionary important foodborne microbes that we have been lost over the course of sanitizing the industrialized environment. This paper that was published online just a couple of months ago actually showed the importance of our original diet. And this is a paper you don't see often, a paper in Nature Medicine from Africa. It's actually based on an extremely robust trial that was done in Tanzania, in the Kilimanjaro region. And they tested the Western diet. You can see the examples of this with sausages and pancakes and fries and ketchup and biscuits. And they compared this to the African heritage diets. With traditional breakfast, with sour fermented milk and taro and stiff porridge, I don't know what that is, green vegetables and okra, and they do it extremely meticulously in both rural and urban dwellers. And in the lower right part, you can see the effects of something, just 1 week of a fermented beverage called memgege, which is a traditional chaga beer made from fermented bananas and millet. And the effects of this traditional African heritage diet was really dramatic. And the switch from the Western style to the heritage style diet or consuming this fermented beverage have anti-inflammatory effects. And I think some of these observed changes in the immune and metabolic profiles persisted at follow-up. This is really an interesting approach to address the inflammatory disease of aging. I also like to mention also something else that published in Nature Communication, which is called urolithin, which is a major microbial metabolite that you will find in polyphenolics, especially from pomegranate fruits and berries. It has anti-inflammatory, antioxidative, anti-aging activities. And in this paper, they actually could show that this urolithin exert barrier functions through activation of NRF2-dependent pathways that upregulate the epithelial tight junction proteins. They could show in the clinical part that treatment attenuated colitis and remedied barrier dysfunction. And this leads me to the positive effects of using berries as food as medicine. I have been interested in the effects of blueberries, which we have an abundance of in Sweden. Actually, 1/4 of Sweden is covered by blueberry bushes, so we have a lot of it. And actually, the scientific evidence is quite, I think, robust. This is a paper in Circulation showing that high intake of anthocyanins by berries is associated with reduced risk of myocardial infarction in young and middle-aged women. We even have a 6-month double-blind randomized controlled trial with blueberries showing that they improve biomarkers of cardiometabolic function. The authors conclude that with size effects predictive of 12 to 15% reductions in cardiovascular risk, blueberries should be included in dietary strategies to reduce individual and population cardiovascular risk. Additional benefit of blueberries is that if you pick them themselves, you will get some exercise. So talking about blueberries, what animal like blueberries? Have you any suggestion? Bears. Bears love blueberries. They eat enormous amount of blueberries, and this leads me to hibernation, which I think is an extremely interesting state in which not only bears but other hibernating animals are totally protected against this disease of aging. And we have been conducting studies on bears for many years. Here we transported one of our CT scans from our hospital up to the northern Sweden, and we conducted examination of the bones of bears. Despite that they are sedentary for 4 to 5 months, they never developed osteoporosis. Here, compared to the immobilized sheep, the bone structure, to the 1-year and 18-year-old grizzly bears, bears are totally protected against osteoporosis despite a sedentary lifestyle. This is a paper also showing that they are totally resistant to muscle atrophy despite that they are sleeping for 4, 5, 6 months. If you and I would go to sleep for this period, we would lose 90% of our muscles, but bears don't do this, possibly due to upregulation of NRF2. I just like to present and kind of stumbled into something very interesting when we were interested in TMAO levels in bears. And we compared data to lions and tigers, carnivores, having a lot of red meat to see the TMAO levels. And then we found, to our astonishment, there was absolutely no TMAO generated in the bears. Instead, they generated massive amounts of betaine. And betaine is something that Paul mentioned that I find extremely interesting. And to understand betaine, you really need to know what it is, the reason why animals and we produce and have betaine. It's an osmotic protection, it protects against oxidative stress, it's involved in the methylation processes, it's supporting liver function and protein synthesis, and of course many papers shows that this is an activator of NRF2 key pathway. And then this gets even further more interesting with this recent Cell paper actually showing, and they had exercising young healthy males, and they could show that kidney-derived betaine during exercise actually act as an inhibitor of this tank-binding kinase, TBK1, and it has potent anti-inflammatory and geroprotective activities. Irrespective if you look at senescence, complements, triglycerides, antioxidants, genomic instability, everything was improved via betaine. So if you are a couch potato like me, you would really like to have something, an exercise mimetic, something you can do instead of exercise. And I think future studies would show if food that is rich in betaine, such as beets and spinach, quinoa, eggs and salmon, may be something that could improve longevity. In this study, we also were interested to see if bears that were not in the natural environment, but captive bears, what happened to their TMAO levels. And in contrast to the free-ranging brown bears, the captive brown bears generated TMAO. And instead, they had about half the levels of the betaine. And we then were interested in captive bears, and if you want to study captive bears, you have these bile farm bears in Vietnam and China, and these are really, really poor bears. That they are living in extremely poor conditions, served crappy food, and these are the cagers, and of course the bile that is extracted from them is used in traditional Chinese medicine. And we could actually show in this paper that in contrast to the free-ranging bears, these bears in captivity, they develop all these burden of lifestyle diseases. They have hypertension, they develop kidney disease, they have chronic inflammation, left ventricular hypertrophy, obese sarcopenia, and developed also joint disease. So finally, I believe that we really need a radical transformation of the global food system. If we could move into more locally produced plant-based diet with more grains, fermented nutrients, and limit our intake of red meat, there is no doubt that this would reduce the risk of burden of lifestyle diseases. And there are a solid, robust evidence for this. Now, these are 2 recent papers in Nature Medicine, Nature Food, showing the optimal dietary patterns for healthy aging, and this paper in Nature Food showed that you can add 8 to 10 years of your life if you shift toward healthier diets. So that's a no-brainer. But on top of this, if you can do a radical transformation of the global food system, you will also reduce the greenhouse gas emissions dramatically. Red meat production is the major cause of greenhouse gas emissions on our planet. I think it's also likely that with such a shift in diet, you can increase the resistance against the environmental stress, such as air pollution. There are already discussions if plant-based diet in areas where you have a lot of air pollution via activation of NRF2, for example, could reduce the toxic effects of air pollution. Air pollution kills more than 7 million people, according to WHO. So here you have something quite unique. You have a win-win-win situation if we can transform our global food system. So the power is on your plate. And with this, I'd like to thank you for your attention.